My name is Kevin Costa, and I'm the Administrative Director for the Synthetic Biology Engineering Research Center here at UC Berkeley. And the goal of the center is to engineer biology, basically, to make it so that we can reduce to practice the engineering of biology. And hopefully that will sort of result in all kinds of benefits for medicine, energy, and information technology, a number of fields. My role as the administrative director is essentially to provide an interface to the NSF. There are many reporting requirements and documentation that they request from us. And I also negotiate with the other Synberg universities at MIT, Harvard, UCSF, Stanford, and Prairie View in order to distribute money across the project and to ensure that the work is proceeding as it should.
So how did you come to be interested in synthetic biology, as in what was your trajectory to your current role?
The way that I became interested in synthetic biology is really just by virtue of being associated with Jay Kiesling. I worked at Berkeley Lab for a number of years as another type of research administrator. And I participated with Jay in some of the projects that he was doing there at Berkeley Lab. And that was when he established the world's first synthetic biology department. And so that was a fun time. We also helped to organize the Synthetic Biology 2.0 conference at UC Berkeley in around 2006. And so I just came to understand synthetic biology from an administrative background that way. I also studied biology as an undergraduate, and so and I kind of am a geek and play with toys and computers and things. So synthetic biology is kind of a nice combination of a lot of those interests.
How would you describe synthetic biology?
Well, the way that I would describe synthetic biology is kind of like the Legos of biology. Think of, little red Legos and blue Legos and yellow Legos, and you just put them all together. You know exactly how they're going to behave and what kind of a part you need to interact with another kind of part to build a specific object. Right now, biology is very artisanal. So if you want to build something in a laboratory, you really have to know your bug, whether it's E. coli or yeast or some other microorganism. You have to know all of the nuances and all of the details of the bug. You have to know the exact gene pathway and how the side products might behave differently in different environments and conditions. And so synthetic biology, if successful, will sort of take away all of those details and black box them, if you will, into these little Lego parts that you can assemble and share with your friends over at other universities if they need a yellow Lego piece to act as a chemical reporter. Then they can just download the sequence for that part from a database, and we'll have this kind of free open source exchange of biological parts.
So what is that organization that manages the parts?
So the primary way that we organize parts now is through the Registry of Biological Parts. And this is maintained at MIT. It sort of pre-existed Synberg, but we sort of support the continued growth and development of it. And it's basically it kind of has two sides. For each biological part, there's the informational side, and then there's the purely sequence side. So you have the description of what the part does, how it reacts, and this is something that we're always trying to develop, is how do you characterize the part in a way that's meaningful for another scientist at another university doing a completely different project? What are the sort of parameters that are going to be interesting to that researcher or any other researcher? And then you've got the sequence in there, and that involves, you know, just the A's, the G's, the T's, and the C's, and how they go together, and also ensuring that in fact that's the right sequence. So through the registry, as we call it, you know, there's an open source resource for researchers to go in and try to find a part of interest through meta tags and descriptions and sequence searches. At least I think they can search via a sequence, I'm pretty sure. Not something I do on a daily basis.
Yeah, I think we already discussed Synbio. What is IGEM?
Oh. So IGEM is it's an undergraduate summer research program. It basically brings together teams of undergraduates at universities throughout the world, really. And they compete over the course of the summer to create just as interesting a synthetic biology project as they can conceive and execute. The projects proceed over the course of the summer and culminate in November at MIT in the IGEM Jamboree. The teams get together and they present their projects, and they compete for prizes such as Best Part or Best Characterization, Best Application, things like that. So it's actually a great opportunity for the students to see how other undergraduate teams have approached the problem, what kinds of what approach they use to the registry in using the parts and putting them together in a kind of clever or creative way. Also, one of the other big byproducts of IGEM is that it's essentially creating a community of young researchers. It'll be the next generation of biologists, and we kind of consider that one of the big outputs of IGEM and Synbio is this young community, or this next generation of researchers who really understand synthetic biology from a conceptual level, and it's kind of in their DNA, if you will, how to engineer parts in a sort of abstract engineering sense.
So, are there any difficulties you anticipate with dealing with the IGEM project on your end?
So the difficulties that we encounter with IGEM are really mostly logistical. You know, putting together the paperwork to get all of the kids together. You know, we're usually bringing undergrads not only from UC Berkeley, from internal, but also from across the country. There's a combination of students that come together. Getting their authorization to work in the laboratory. Some of them are minors. You know, getting their travel together, getting the funding to run the team. It's actually quite expensive, depending upon who you ask. It costs $25,000 to maybe $100,000 just for the summer to run a team of 8 to 12 students. So there's a lot of logistical stuff that has to happen. Here at UC Berkeley, the Berkeley IGEM team is fortunate enough to have some really excellent instructors, such as Chris Anderson and John Duber.